Welcome students. I'll start this session with an important statement. Fat burns in the flame of carbohydrates. What do I mean by this? Now if you look at it, I suppose I've got a lamp and I've got a wick. I try to burn the wick. What will happen? The wick is made of cotton. Naturally, if I burn the wick is going to get burnt within no time. But if you look at the liquid or the oil that is present in a lamp, and if you try to burn it using a candle or something, that liquid is not going to burn. But if you are going to put that wick into the lamp and then you are going to light the wick, what will happen? The lamp is going to be lit for a longer time. And what is this? What is that oil in it? It is a lipid. What is the wick? Wick is a carbohydrate. So for the burning of that oil, we need a wick. Only then it will give out that energy. It will burn, it will get oxidized. So for the oxidation of the lipid, we need a carbohydrate. Is this happening only in the candles, in the lamps? Candle also has a wick. No, it is not just restricted to outside the body, in vitro. It is happening even within the body. Even within the body, for the burning of fats, we need carbohydrates. So we call this as fat burns in the flame of carbohydrates. So can we justify this statement within our body? Can this really happen? Yes. What is that carbohydrate and what is that fat? So for the oxidation or burning of fats, carbohydrates are essential. The wick is essential. And why is it? Now, if you look at the fat, lipid, from where has it come? Either from the adipose tissue. Adipose tissue has undergone lipolysis, released fatty acids. These fatty acids are undergone beta oxidation and released acetyl-CoA. And this is the acetyl-CoA which has to enter into the TCA cycle. To enter into the TCA cycle, what does it require? It requires oxaloacetate. And from where has oxaloacetate come? From pyruvate. From where has pyruvate come? From glucose. So what is the source of oxaloacetate? It has come from carbohydrates. And from where has acetyl-CoA come? Mainly, partly from, I would agree, from carbohydrates. But if more of it is coming from beta oxidation. So it has come from lipids. So acetyl-CoA is the end product of fatty acid oxidation. And this has to enter the TCA cycle. For this, it has to combine with oxaloacetate, and oxaloacetate is derived from pyruvate, which has come from glucose. So, the entry of acetyl-CoA into the TCA cycle is dependent on the availability of oxaloacetate. Suppose there is a condition in which the amount of acetyl-CoA is more than it can enter into the TCA cycle. What will happen? This acetyl-CoA is going to get diverted. This acetyl-CoA needs to have another pathway. So, fat and carbohydrate degradation has to be balanced. The amount of acetyl-CoA has to have enough oxaloacetate, only then they can get oxidized. So, if at all fat breakdown predominates, that is, there is more of acetyl-CoA, then acetyl-CoA has an alternative fate. So, acetyl-CoA, I write it here, acetyl-CoA, now it cannot enter into the TCA cycle. Why? There is no oxaloacetate. It is not able to enter. It goes into a alternative pathway, and that is ketone bodies. Ketone bodies. So the main topic for this session is ketone bodies. Acetyl-CoA forms ketone bodies. Ketone bodies are also called as acetone bodies. It involves three main ketone bodies: acetone, acetoacetate, and beta hydroxybutyrate. These are the three main ketone bodies. So let us look at ketogenesis. Where is ketogenesis is taking place? Ketogenesis is formation of ketone bodies. They are formed from acetyl-CoA. Naturally, when acetyl-CoA is more, they are formed in the liver, and in the subcellular matrix is the mitochondria. Now, which are the enzymes and the pathway? Let us look at ketogenesis. As I said, ketone bodies are formed when acetyl-CoA levels are more. So, we have acetyl-CoA is more. So 2 molecules of acetyl-CoA will combine to form release one coenzyme A and form acetyl-CoA. Now in the next step, this is catalyzed by the enzyme. Please look here. Beta ketothiolase. Beta ketothiolase is responsible for the combination of 2 acetyl-CoA to form acetoacetyl-CoA. In the next step, the next enzyme is HMG-CoA synthase. So in this, acetoacetyl-CoA combines with one more molecule of acetyl-CoA, to form what is called as HMG-CoA. HMG-CoA stands for beta-hydroxy-beta-methyl-glutaryl-CoA. Accepted abbreviation is HMG-CoA. You need not worry about the full form of HMG-CoA. So one more molecule of acetyl-CoA is released and it forms HMG-CoA. The enzyme is HMG-CoA synthase. Now in the next step, HMG-CoA, where have you heard of this? This whole thing till here. The similar thing is also happening in the cytosol, and HMG-CoA is the initial steps that are happening in the cholesterol biosynthesis also. The same thing is happening in cholesterol biosynthesis, but here this pool of acetyl-CoA is different, that pool of acetyl-CoA is different. This is happening in the mitochondria, that is happening in the cytosol. And this HMG-CoA synthase is different, that HMG-CoA synthase is different. The further fate of HMG-CoA is also different. Remember, in the cytosol, it is acted upon by HMG-CoA reductase to form further actions. So HMG-CoA reductase is the regulatory enzyme of cholesterol synthesis, whereas here it is another enzyme, HMG-CoA lyase. HMG-CoA lyase will release acetyl-CoA and acetoacetate. It gets lyase, HMG-CoA gets broken down, releases acetyl-CoA which can be reformed and forms acetoacetate. What is acetoacetate? This is the primary ketone body, what we call as the primary ketone body. Acetoacetate can now be converted, can have two fates. One, it can be converted to beta-hydroxybutyrate. Beta-hydroxybutyrate. What is this beta-hydroxybutyrate? It is the secondary ketone body, and it can be spontaneously, non-enzymatically undergo decarboxylation, loses its carbon dioxide to form acetone. Now, beta-hydroxybutyrate to acetoacetate, it requires NAD. So, in this NAD, the enzyme is beta-hydroxybutyrate dehydrogenase. So, acetoacetate for its conversion requires NADH is converted to NAD+. This is a reversible reaction. Now, whether acetoacetate is more or beta-hydroxybutyrate is more, all depends upon the NADH, NAD+ ratio within the cell. If NADH is more of beta-hydroxybutyrate is formed. If NAD+ is more of acetoacetate is formed. You have to just look at this reaction, and which one is more and how to find out is the enzyme is beta-hydroxybutyrate dehydrogenase. So, dehydrogenase means NAD+ has to get hydrogen. This is beta-hydroxybutyrate dehydrogenase, so it gets converted to NADH. So this is the meaning of this is a reversible reaction. If NADH is more, beta-hydroxybutyrate is formed. If NAD+ is more, acetoacetate formation is more. So depending upon the redox status of the cell, which is the predominant form will predominate, and it can just undergo spontaneous decarboxylation to form acetone. So let us look at the different things. As I said, the ratio between beta hydroxybutyrate and acetoacetate depends upon the mitochondrial NADH-NAD ratio. When NADH is more, when is NADH more? Whenever the energy status within the cell is more, beta hydroxybutyrate increases. If the electron transport chain is functioning at maximum capacity, then NAD plus will increase. At that time, it will result in the reverse and acetoacetate becomes more. So it all depends upon the redox state of hepatocyte. Suppose it is alcoholism which has led to ketone body formation, or suppose there is alcohol intake, for example, what will happen? Alcohol during its metabolization will, by alcohol dehydrogenase, will produce NADH. This NADH will increase. If NADH increases, it results in the formation of beta hydroxybutyrate. So it all depends upon the redox state of the cell. So why are they formed and how are they utilized? Please remember that acetoacetate, beta hydroxybutyrate or acetone, more than that, these two are water soluble. There lies the whole thing. Now you had fatty acids which were lipids. So we are now converting a lipid into something which is water soluble and freely diffusible. So ketone bodies are water soluble. They diffuse out from the liver mitochondria into the blood and are transported to various tissues. Now what is happening in different tissues? Let us look at what is happening in different tissues. Actually the reverse of this has to take place. So let us look at either beta hydroxybutyrate to acetoacetate is formed. Now the reverse is possible, isn't it? Now this is spontaneous and this is non-reversible. Formation of acetoacetate from acetone does not take place. So let us look at beta hydroxybutyrate or acetoacetate. Reversible reaction, they are formed. Beta hydroxybutyrate dehydrogenase. Now the next reaction is catalyzed by the enzyme thiophorase. It has to get activated. It has to form acetyl-CoA. So something has to donate its coenzyme A, and this coenzyme A is donated by succinyl-CoA. Succinyl-CoA combines with acetoacetate to form acetoacetyl-CoA. The enzyme is thiophorase. This acetoacetyl-CoA enters into the beta oxidation. Could be beta oxidation and then give out energy if it is required. We are talking of a reverse reaction. So we said that acetyl-CoA is converted in this direction because you cannot have TCA cycle happening. But this is happening in the liver. Now let us talk of other tissues. These ketone bodies have already been transported to other tissues. Okay. So where gluconeogenesis is not taking place. In the liver, that oxaloacetate has gone for gluconeogenesis. So availability of oxaloacetate has decreased. But in other tissues, oxaloacetate levels have not decreased. So this acetoacetyl-CoA can be broken down to form acetyl-CoA. And this acetyl-CoA, this enzyme acetoacetyl-CoA2, this is a part of beta oxidation. Acetyl-CoA enters into the TCA cycle. Okay, in other tissues, please remember this is all happening in other tissues except the liver. This enzyme thiophorase, which is responsible for the utilization of ketone bodies, this enzyme is absent in the liver. It doesn't make sense for the liver to synthesize these ketone bodies and utilize these ketone bodies by a mechanism which is not happening. The TCA cycle is hampered in the liver. Why? Because oxaloacetate has been diverted. So in other tissues this can take place, but not in the liver. So the liver cannot utilize ketone bodies. Ketolysis does not take place in the liver. So let us look at this. Enzymes thiophorase, thiolase to form acetyl-CoA which can enter into the TCA cycle. So tissues which cannot utilize ketone bodies include liver because the thiophorase enzyme is absent. Are there any other tissues? Yes. Which are they? Remember, this whole thing is happening in the mitochondria. What if a tissue, what if a cell does not contain mitochondria? And can you give an example? Yes, the erythrocytes. If you're told erythrocytes, very good. Erythrocytes and RBCs and other tissues which lack mitochondria also cannot utilize ketone bodies, which lack mitochondria. So to summarize, ketogenesis allows the liver to oxidize increasing quantities of fatty acids. More of beta oxidation can also take place, so it will get its energy that the liver requires from fatty acids. It will get through beta oxidation. At the same time, this ketone body formation will see to that its TCA cycle is not hampered, or it's more than the TCA cycle, its gluconeogenesis is not hampered. So for gluconeogenesis it requires energy, and that comes from beta oxidation. And at the same time, if it had gone, if all this oxaloacetate had gone for TCA cycle, gluconeogenesis would have been hampered. So at the cost of this gluconeogenesis, for the sake of gluconeogenesis, it has an alternative pathway and it can oxidize fatty acids and help in the formation of ketone bodies. Please remember, acetoacetate and beta hydroxybutyrate are used by extrahepatic tissues as a source of energy. Heart muscles and renal cortex prefer ketone bodies to glucose, and in case of starvation and diabetes, brain also utilizes ketone bodies, sparing glucose for the tissues which have absolute requirement for glucose, like the RBCs. So in case of starvation and diabetes mellitus also, the brain utilizes ketone bodies. So let us look at how this whole thing is regulated. Regulation of ketogenesis occurs at 3 levels. One, at the level of adipose tissue. Remember, adipose tissue has to undergo lipolysis. Secondly, at the level of entry of fatty acids into the mitochondria for beta oxidation. And thirdly, at the level of entry into the TCA cycle, acetyl-CoA to TCA cycle. So at the level of lipolysis, it is hormone-sensitive lipase, insulin inhibits, glucagon stimulates. At the level of mitochondria, it is carnitine acyltransferase 1, which is inhibited by malonyl-CoA, and at the entry, it is depending on the availability of oxaloacetate. So let us look at the clinical significance of these ketone bodies. Now, normally, some amount of ketone bodies are produced and some utilized, so that the concentration of ketone body in the blood is less than 1 milligram per dl. Always, this is normally happening. Some amount is formed, some amount is degraded. Ketogenesis and ketolysis. But the amount of ketone bodies is less than 1 milligram. But when the rate of synthesis exceeds the rate of utilization, it can lead to ketonemia, means increased ketone bodies in the blood. Ketonuria, they are water soluble, so they can be excreted into the urine. Ketonuria, plus the smell of acetone in the breath. The breath, because acetone is volatile, it gets exhaled out. Acetone has got a fruity odor. So these three together, ketonemia, ketonuria and smell of acetone in breath, together form the condition called as ketosis. When I say ketosis, it is actually misnomer. I'll talk more about ketosis as we go ahead. What are the causes for ketosis? One important cause is uncontrolled diabetes mellitus or prolonged fasting and starvation. Let us look at how ketosis is got in these two conditions. So in diabetes mellitus, most common cause, there is deficiency of insulin. Glucagon is increased, there is deficiency of insulin, glucagon is increased, so increased lipolysis takes place. Remember, the adipose tissue is starving of glucose, the glut transporter 4 is not gone, and the adipose tissue is thinking that there is no glucose, so it undergoes lipolysis, so increases the free fatty acids leading to increased oxidation and acetyl-CoA. At the same time, because of the imbalance between insulin and glucagon, because of the lack of insulin, gluconeogenesis is stimulated, oxaloacetate is diverted. So acetyl-CoA cannot enter TCA cycle, it is diverted to ketone body formation. Now in starvation also the same thing is happening. Now there is one more condition which is hyperemesis, also I have written, especially involved in the first trimester of pregnancy. During this there is a lot of vomiting, but more than vomiting there is no glucose intake. In these cases also sort of condition which is similar to starvation is happening. Remember, whenever blood glucose levels are low, glucagon is high. So what does it do? Dietary supply of glucose not there. Glucagon causes lipolysis to meet the energy needs of the body. So there is increased fatty oxidation, increased acetyl-CoA. Oxaloacetate is diverted to gluconeogenesis. Acetyl-CoA cannot enter and diverted to ketone body. And ketone bodies are used as a source of fuel by the brain. That is the main reason in starvation we have ketone body formation to act as a source of energy to the brain. In diabetes mellitus, no such thing is happening. Why? Because the brain does not require insulin for the entry of glucose. So glucose is high, the brain keeps on utilizing glucose only. It is only in starvation that the brain switches over to the use of ketone bodies. So main thing is oxaloacetate is diverted to gluconeogenesis. Now let us look at the effects of ketosis. It is also called as metabolic acidosis. Why? The ketone bodies acetoacetate, beta hydroxybutyrate are actually acids. So they are called as keto acids. The right word would be ketoacidosis because the acids levels have increased. Whenever acid increases, the plasma buffers and the bicarbonate level decreases. There will be Kussmaul's respiration and hyperventilation. There will be smell of acetone in the breath, and plus there will be osmotic diuresis because when this ketonuria is going out of the body, it will drag water along with it. So there will be osmotic diuresis. This can lead to dehydration. It can lead to loss of electrolytes like sodium and potassium, lead to hypokalemia, lead to hyponatremia, finally leading to coma or death. So it is important that you know about ketosis, dehydration, coma and death. Now, which are the lab tests that are done? Include blood glucose, we look at acid-base parameters, we look at serum electrolytes. As I said, sodium and potassium can also be dragged along with either acetoacetate or beta hydroxybutyrate, and urine for ketone bodies, which is done by the Rotheras test. So, I'll just give one thing about it. If a Rotheras test is positive in the urine sample and Benedict's test is also positive, can you comment on the clinical condition? And I want you to pause your presentation here and look at this slide. What do you think it is happening? And this is one more condition in which the Benedict's test with the urine is negative. Here Benedict's test is positive. So what are your comment on the clinical condition? Please look at it and try to think over the answer. If Benedict's test is positive, it means he is having glycosuria. Most probably this person is having uncontrolled diabetes mellitus. If Benedict's test is negative, it means his urine is not containing glucose. This might be mostly starvation induced ketoacidosis. This might be this is uncontrolled diabetes mellitus associated ketoacidosis. So with this we come to the end of the ketone bodies, how they are formed, how they are utilized, and what is the clinical significance of these ketone bodies. Thank you.